Ligating a PCR product into a vector with topoisomerase. Cloning system with topoisomerase. Topoisomerase 1 base cloning explodes the reaction depicted in the following figure to join DNA fragments containing 5' hydroxyl groups to a substrate plasmid to which the Bacina topoisomerase 1 enzyme is covalently attached. Therefore, Bacina DNA topoisomerase 1 shared vectors known as topovectors are classified into topo-TA and topo-blunt vectors. Topo-TA have sticky thymine, which are routinely used to clone polymerase chain reaction (PCR) products, with an extra adenine at their 3' end, while topo-charred blunt vectors are used to clone blunt end of PCR products. PCR 2.1 topo vector was designed to accept PCR products made with tag DNA polymerase, and therefore having the 3' overhanging of adenine nucleotides. As they sail it, the vector is already linearized and has 3' overhanging thymine residues, and it's activated by having a Bacina virus topoisomerase 1 third to HN. Because of the kinetic advantages of having the topoisomerase enzyme on a leash, the reaction only takes 30 seconds to 5 minutes to run, compared to a typical ligation reaction that might be run overnight. The PCR 2.1 topo vector is 3.9 kilobases long and has a lag theta A fragment, an M13 reverse priming site, a multiple cloning site, a T7 promoter priming site, M13 forward priming site, F1 origin, kinemosin resistant and ampicillin resistance, and ERF and a PUC origin. Ligate. The topo vector has an overhanging of 3' thymine. Your PCR product must have an overhang of 3' adenine. Be sure to include a final extension step of 7 to 30 minutes during the PCR. Longer PCR products will need longer extension times. Among the various applications of the PCR 2.1 topo vector, here are two main applications: selection of competent cells for high-speed electroporation or routine cloning, and high-level protein expression using E. coli. Ligation involves joining of the ends of DNA with other ends. However, each DNA fragment has two ends, and if the ends are compatible, a DNA molecule can circulate by joining its own ends. At high DNA concentration, there is a greater chance of forming intermolecular ligation, while at lower DNA concentration, intramolecular reaction that circulates the DNA is more likely. The transformation efficiency of linear DNA is much lower than circular DNA, and for the DNA to circulate, the DNA concentration should not be too high. As a general rule, the total DNA concentration should be less than 10 micrograms per milliliter. The higher the ligase concentration, the faster the rate of the ligation. Blunt end ligation is much less efficient than sticky end ligation, so a higher concentration of ligase is used in blunt end ligations. High DNA ligase concentration may be used in conjunction with PAG for a faster ligation, and they are components often found in rapid ligation kits. Two issues are involved when considering the temperature of ligation reaction. First, the optimum temperature for DNA ligase activity, which is 37 Celsius, and second, the melting temperature of the DNA ends to be ligated. The ionic strength of the buffer used can affect the ligation. The kinds of cations present can also influence the ligation reaction. For example, excessive amount of sodium causes the DNA to become more rigid and increase the likelihood of intermolecular ligation. At high concentration of monovalent cations, greater than 200 millimolar, ligation can also be almost completely inhibited. The standard buffer used for ligation is designed to minimize the ionic effect. For screening a close containing recombinant DNA, a chromogenic substrate known as X-gal is added to the agar plate. If beta-galactosidase is produced, X-gal is hydrolyzed to 5-bromo-4-chlorohydroxyl, which spontaneously dimerizes to produce an insoluble blue pigment. The colonies formed by non-recombinant cells therefore appear blue in color, while the recombinant ones appear white. The desired recombinant colonies can be easily picked and cultured. IPTG is used along with XGAL for blue-white screening. IPTG is a non-metabolizable analog of galactose that induces the expression of the LAC-STAT gene. It should be noted that IPTG is not a substrate for beta-galactosidase, but only an inducer. For visual screening purposes, chromogenic substrate like XGAL is required.
No one will appreciate the colonies in the picture, but the growth of blue and light blue cells white is observed, which indicates that there are bacteria in which the plasmid cannot quite link it with the PCR product, and there are colonies in blue. If he can't link the plasmid PCR with the color of the colonies are white. And finally, mine in the case of light blue colonies may not respect it is the open rate frame of the gene beta-galactosidase. In the next chart, we can see the colonies blue are 6, white 22, and white and blue are 20. In the next picture, we can observe the blue boxes are white colonies which are transformed bacteria, and in red box the blue colonies which are non-transformed cells are shown. Transformation efficiency. The efficiency is calculated by UFC colonies forming units per nanogram of DNA plus 1000 nanograms per 1 microliter plus the dilution factor. The efficiency of this practice was 9.6 by 10 to 3 UFC plus micrograms. The ligation reaction was carried out using the vector PCR 2.1 topo, which is probably linearized with topoisomerase 1 is ready to ligate PCR products. The PCR product was previously amplified with Taq polymerase, so the DNA sequence were left with 3' overhands compatible with the topo linearized plasmid that has 3' T overhands. The compatible PCR product and vector were then ligated by topoisomerase 1 ligase activity, which then realized itself from the DNA. The vector PCR 2.1 topo is provided with the ORI that is a high copy numbers original replication is useful for selecting the transformer cells in culture. It contains a LAC-A alpha that encodes for the beta-galactosidase, a fragment useful from the XGAL identification. It has the resistant genes from ampicillin, neomycin, and kanamycin. These are used for selection, and the CAM binding sites this involves in the transcription of several genes.
When cloning PCR-blind products, Thermo Fisher offers a solution, zero-blind topo technology, which is highly efficient and quick. It only takes 5 minutes. Also, they offer PCR4 topo TA vector for direct ligation of Taq-amplified PCR products in just 5 minutes and yielding up to 95% of recombinants. There exist different ways of screening, which include the following. Blue-white screening, a popular technique which consists of the insertion of a gene into a vector containing a LAC sequence. Positive selection vectors express a lethal gene, and when the insert has been cloned into the vector, it disrupts the multiple cloning site and only cells with recombinant plasmids are able to grow. Sequencing, the most accurate way with appropriate sequencing primers and Sanger sequencing method. Emphasizing the color screening, it is not 100% reliable, because this method can throw false positives due to the possible ORF being undisrupted. As for the efficiency, it can be transformed cells with linear fragments, but the efficiency is lower. Therefore, it is highly recommended to use supercooled plasmid because it has a higher efficiency and also it is quicker. On the market, top-type cells contain different features in their genotype. For example, AR14 blocks arabinose catabolism, NA eliminates non-specific endonuclease activity, and LAC-C the beta-galactosidase gene, which yields colorless colonies in presence of X-gal, among many other things. Top-down cells utilize different topobacters depending on your needs.
As any protocol to follow these steps well and report time is extremely important for obtaining more reliable results. Now, the very small volumes of reagents are used is when you should have the proper pipetting techniques to avoid loss and take appropriate aseptic techniques to avoid contamination.